So I'm Shaheed. I received my PhD on the East Coast of the United States at the University of Pennsylvania, and now I work on the West Coast of the United States at Oregon Health and Science University. My training is as a cellular immunologist, and I know this is not an immunology conference, so I'm going to try to give a little bit of background to ground everyone in what I exactly do. So I specifically study pathogen-specific CD8 T cells, and you really can't understand a CD8 T cell without understanding the major histocompatibility complex, or MHC or HLA, if you're talking about humans. So this molecule has one specific purpose. It's expressed in almost every single cell in the human body, and what it does is when a cell is either bacterially or virally infected, that infected cell breaks down that virus or bacteria into peptides or into epitopes that can be presented on the MHC molecule. You can think of it kind of like a key when you combine these two components together. And this key can unlock a CD8 T cell through its T cell receptor. And once unlocked, that T cell can kill that virally or bacterially infected cell. And one thing to note here is typically this is understood as a fairly binary interaction. There is one key for one lock, one epitope for one CD8 T cell. So you can imagine that such a binary system, when you're talking about the entirety of the human population, needing to deal with so many different pathogens, that you would need a massive amount of diversity, and that is what this system generates. If you look amongst the entire human population, we have approximately 11,000 unique MHC molecules that are expressed amongst us. Of course, no one individual has all of these molecules. We only express about 6 of them if you're talking about canonical MHC molecules. Now, as to why that is important, that means each and every single last one of us generates unique epitopes in unique CD8 T cells. And as to ground this into something that we would all care about, we can think of HIV elite controllers. These individuals are blessed by having specific MHC molecules that present the correct epitopes to CD8 T cells, allowing these individuals to not only control their virus below the point of detection, while most people don't particularly have these MHC molecules and thus don't have these CD8 T cell responses. So how does this come back to what I specifically do at my institute? Well, my institute has done a lot of work trying to develop rhesus CMV strain 68-1 as a human clinical vector. In our preclinical work using the rhesus monkey model, we've shown that this vaccine vector can protect over 50% of rhesus monkeys from SIV infection. This vaccine is unique, though, in that it doesn't generate SIV-specific antibody responses, the antibody responses we heard about earlier today, but instead what it does generate are CD8 T cell responses. Now, these CD8 T cell responses are fairly unique. Here we're just looking at several vaccinated animals, and I'm not going to go into a lot of detail here, but what I do want to point out is each one of these boxes are one of those epitopes I was telling you about earlier. So what this vaccine does is induces a wide breadth of T cell responses. The other thing that it does is it generates 2 different flavors of these T cell responses. Either they recognize keys in MHC class 2 form or on MHC-E. Now, MHC-E is kind of interesting. Unlike the molecules I was telling you about before, with that high amount of diversity, 11,000 particular molecules across human beings, MHC-E is incredibly conserved across all primates. And when looking at humans, we only have 2 functional alleles of this protein. So this led us to an interesting question. So if you're talking about only one particular key system, how do these CD8 T cells function? How do they garner all this breadth? How do they work? So we did many different assays, and what I am going to show you is our SIV-infected recognition assays. So here, we take CD4 T cells, infect them with SIV, and we use those to unlock CD8 T cells from a vaccinated animal. And from there, we can analyze those T cell responses and pull out those T cell receptors I was telling you about. And here we see that these animals, this particular animal, and all the animals that we've studied, only generated a relatively small set of T cell receptors. So what we're actually seeing here is that we have multiple different keys for basically one lock, which is in contrast to our understanding of how CD8 T cells are supposed to function. We moved forward to prove this by taking these same T cell receptors and introducing them into CD8 T cells from animals that never saw the vaccine, so we could specifically look at what these T cells do. And what we noted was what we saw from our sequencing, that these particular TCRs are capable of recognizing multiple different epitopes, breaking our understanding of how CD8 T cells function. And I'm being told I'm out of time, and that's great because that was my last slide. And I would just like to thank you all for your attention, and I would also like to thank all our wonderful collaborators who've helped on this project. Thank you.